Hi, Gen Bio. Dr. Fry here. I am here to give you the introduction to week 2 biology lab, which covers echinoderms and chordates. Okay, so these are both deuterostomes. Okay, deuterostomes, like I said, include echinoderms and chordates. And the organisms that we're going to look at in those groups are the sea star, which is an echinoderm, and we have a couple examples of chordates for you. We have the Amphioxus, which is also called a lancelet. We have the lamprey, which is we have the adult form, which we're going to look at on the slides here, and we have some in lab that you can look at, but we're not dissecting. And then we have the larval form, which we refer to as Amicetes, or I've heard it pronounced Amicoetes. I'm not sure how you pronounce it. Your guess is probably as good as mine. And then we also have the perch, which is a type of ray-finned fish, which we'll have videos for you to view. And when you return to campus, hopefully you'll be able to dissect that also. Okay. So like I said, these are all deuterostomes. And deuterostomes, if you just will turn your attention to the phylogenetic tree, are circled here in red. If you recall from the last week's material, we had looked at protostomes, which are over here. They're closely related in that they're both eumetazoans. They both have bilateral symmetry. They both have body cavities. And now we branch off. Okay, so last week we looked at protostomes, and this week we're looking at deuterostomes. So last week I had detailed a few of the differences between protostomes and deuterostomes. So some of this should look familiar in that we had talked about the formation of the coelom, that space between the digestive tract and the body wall, and how that forms. And I'd also mentioned that the fate of the blastopore is different between protostomes and deuterostomes. So for protostomes, the mouth is the structure, the digestive tract structure that develops from the blastopore, versus in deuterostomes, it's the reverse. The anus is what develops from the blastopore, and then the mouth is the second opening as that tube for the digestive tract forms. Okay, now I'll mention one more difference between protostomes and deuterostomes this week. I don't believe I mentioned this last week, but they also have a difference in how cleavage happens. So cleavage, if you recall, is the early developmental stage where there's a lot of cell division, but not much cell growth. So you've got one cell turning into two, two turning into four, four turning into eight, and so on. And as each of these divisions happens, we get a ball of cells, right? Well, in protostomes, the cleavage furrows, the formation of different cells, occurs in such a manner that the top four cells in the 8-cell stage, the top four cells are almost diagonal. They're spiral, they're catty corner to the bottom four cells, okay, as this division forms along the midline here, okay? So the top layer of cells are usually smaller, and they sit sort of in the grooves between the bottom two cells, okay? And that looks different in deuterostomes, where we have that spiral orientation, that diagonal orientation. In the protostomes, we see a more linear orientation. So the top layer of cells sits pretty much directly on top of the bottom four layers of cells when we're talking about the 8-cell stage, okay? So that's called radial cleavage, all right, as opposed to spiral cleavage. And also you'll notice here that at this stage, protostomes are listed as having determinate cleavage. And what this means is that the fate of each of these individual cells is determined very early on. So if you were manipulating this ball of cells and you removed one, you would produce an organism which is missing parts. Basically, the fate of these cells is determined very early on in development. Versus in deuterostomes, this is different. The cell's fates are not determined as early. So at this point, if you removed one of these cells, it actually might have the potential to develop into an entire organism. Okay, so its future is not yet determined, and it has the capacity to differentiate still and become different parts of the organism. So this is one feature that allows for humans, for example, to have um, identical twins, because after fertilization occurs, you can still have division. It's rare, obviously, but the individual cells can still continue to divide, and their fate has not yet been determined. So it could potentially produce an entirely new organism. Okay. So that's one other difference that I wanted to point out between protostomes and deuterostomes. Okay. And today, like I said, we're focusing on deuterostomes. Okay. We looked at the protostomes last week. So let's look at our first example of a deuterostome, which is a sea star. Now, you might stop me right here. And if you remember, I had said that we're looking at all organisms with bilateral symmetry. And you're like, now, wait a minute here. Sea stars look as an adult like they have radial symmetry. So as an adult, you could cut them pretty much in any plane. They're mostly symmetric. There is some slight details that would not make them 100% symmetric along any plane as the adult sea star. But as we looked at last semester, they, in the larval form, have a very clear bilateral symmetry. We had looked at their early development on slides last semester, and you saw that they had bilateral symmetry. Okay, so they are characterized based on their early development. As the larval stage, they exhibit bilateral symmetry, whereas as an adult, you kind of say, well, that looks a lot like radial symmetry to me, but they are classified because of how they develop in the larval stage. So we're actually going to just look at the adult, though, for this week. We have videos for you that show the dissection of this organism that's posted on Canvas. You can go through and watch Jen Thornsberry. She's our lab coordinator. She dissected this for you, but I will talk you through some of the anatomy now, but be sure you view that dissection video as well. Okay. So just to start with, we're looking at the external anatomy here. Okay. So there's two different sides to the sea star. The top, or dorsal side, is actually called the abral surface. Okay. And the bottom, the part that kind of sits down towards the surface, the ground, is the oral side. And that's because it has the mouth. Okay. So this is the oral side, and this is the abral side. Okay. So the mouth is right here in the middle. Okay. This little hole, little tiny opening. And then it has 2 feet on this side as well. Pretty easily seen. And then if you go into lab, you can see the actual specimen. You'll see that these 2 feet are a lot softer than all the spines, which are on the outside. Around the 2 feet, there are spines, little prickly projections. And on the abral side, there are a lot of spines as well. Okay. And you'll also notice, I'm going to turn us to the next slide to see it a little bit better, but you'll also notice on the abral side, in addition to all these spines, you've got this one little pale circle here. Okay. This is called the madreporite. And this is a porous structure that allows for water to enter into the sea star. Okay. So as water enters, it's used in what we refer to as the water vascular system of the sea star. It takes in water, and it allows that water to pass through different channels. We'll talk about all of those yet. All of those channels then will allow for the fluid movement throughout the body, as well as help in locomotion for the sea star. Okay, so we'll talk about all of that after I get through some of the external anatomy. Okay, so I did just want to point that out. That's on the abral surface, the madreporite. Of course, you got lots of these little spines and bumps. The center circle is just referred to as the central disc. And then there's usually, on the ones that we have, there are usually 5 arms, okay, or rays that extend from the central disc. Okay. different uh, numbers of arms could be present on different similar organisms, but the ones we get are usually five, usually have five. Okay. Now, if you flip it over and you look at the oral surface now, this is what the oral surface looks like. Okay. So in the very center of the central disc, this is the mouth. Okay, you can kind of see there's a pore there. And I had mentioned that tube feet extend down each of the arms, and they are in what we call the ambulacral groove. Okay, ambulacral groove has the tube feet, all of these. Okay. And then I've included this picture here because it's showing you that this is still the oral surface, and you can see there's like something coming out of the mouth here. This picture here shows that the cardiac stomach, part of the sea star stomach, can actually be projected through the mouth, and this allows for the organism to feed. Okay, so this allows for grasping and digesting of some of the prey for the sea star. Okay. All right, let's move to our next slide, where we're going to look at some internal anatomy then for the sea star. Okay, so if you were to cut off one of the top layers of the sea star, so if you cut sort of down at the tip of the arm and cut up and remove that top layer of spines, then what you would see looks something like this in this first top left picture. Okay, so you can see there's like little grape like bunches that are in each there's two in each arm that extend down each side of each of the arms. These are the digestive glands. Okay, number 8 here shown here is the digestive glands. They are taking up the largest amount of room in the arms. They often extend pretty much all the way down to the tip. Okay, and like I said, there's one on each side, so there's a lot of mass taken up by the digestive glands. Okay. And then if you were to remove those, you would see that there were gonads underneath, and they are shown here. They're paler. This structure here is another sort of smaller bunch of grapes that's covered up by the digestive glands. Okay, but if you can kind of make it out, there's one, here's one, there's one right there. They're in each of the arms as well. They're usually smaller. Sometimes they can get quite large depending on the developmental stage of the sea star, but they look similar, usually paler color to the digestive glands, but in each of the arms as well. Okay, and smaller than the digestive glands. All right, and then we also should look for the stomachs. When you dissect it, you should be aware that there is a pyloric stomach and a cardiac stomach, similar to what we saw in the crayfish. So the pyloric stomach is on the top. It's on the abral side, the dorsal side, just to be more clear. And it has a star-like appearance. Okay, and it's shown here. It's a little hard to make out, but it's some of it was removed with the central disc here, and then some of it is left here in the central disc section. So the pyloric stomach has a star shape. So it has ducts that come and connect the pyloric stomach, which is in the center, to the digestive glands, which are in each of the arms. Okay, so you should look for those. And then underneath the pyloric stomach is the cardiac stomach. And instead of being star shaped, it's more fluffy, like a pillow, kind of like bulbous looking. So this is the cardiac stomach here. Okay. And if you were to remove that, then you'd be able to see all of the structures of the water vascular system. Okay, so the water vascular system we're going to look at on the next slide as well, but I'll mention just so you can see it here. If you remove the stomach and the digestive glands and the gonads, you'll see there's like the madreporite, which we looked at on the last slide as well. That's the entrance to the water vascular system. Okay, and then it is connected through a small tube shown here called the stone canal to the ring canal, which runs along, it's in a circle, a ring, along the entire central disc there. And then from the ring canal, we have a connection to the radial canals, and they go down each of the arms here, indicated by number 11. And then the radial canals are each connected to lateral canals. And the lateral canals reach out from the radial canal and connect the radial canal to the ampulla of the tube feet. So let me move now, just to be a little more clear. Let me move to the next slide to show you all this in um, probably a clearer picture. So the madreporite, again, is on the abral surface. It's the entrance to the water vascular system, and it's connected to the stone canal, which then is connected to the ring canal. That's connected to each of the radial canals, which travel down each of the arms. And then the radial canals are connected to lateral canals, these little guys here, that take the fluid traveling through the radial canal to the tube feet. And the top part of the tube feet is referred to as the ampulla. Okay, ampullae is plural. Okay. So this is the pathway that, like I said, that the water can travel through in order to extend it through the organism and help the sea star with locomotion. So as the pressure from that water enters into each of the arms and travels into the ampullae, then contraction of that ampulla will put some pressure on the tube feet. Okay, so push that water into the tube feet, and this will create a rigidity in the tube feet in that area. And so you can have some rigidity in some areas and simultaneously some flexibility in other areas. And this is what allows for the slow movement that we are familiar with sea stars using for their locomotion. Okay, so that is the water vascular system, the pathway that water takes to travel through the sea star. Okay. So that is, I believe that covers most of the anatomy for the sea star. The one thing I did forget to mention is that at the end of each of the arms, there's an eye spot. I don't think I mentioned this, but at the very tips of the arms, there's a little eye spot which senses light for the organism and allows it to just be aware of how its depth, how much light is hitting it. Okay. All right, so now let's move on to our chordates. So the sea star was an echinoderm, which is a deuterostome, but it is not a chordate. So now we're going to move into the chordates. And there are 4 different characteristics that you should be aware of that are definitive of chordates, okay? And they are the notochord, the dorsal nerve cord, pharyngeal gill slits, and the postanal tail. Okay, so you should also know that these characteristics are not always present at every single stage of life for each of the organisms. So, for instance, we are chordates, but at the moment I don't have a tail or gill slits. But as human development takes place, these structures develop into other things. Okay, so some are lost as development takes place, and at some stages they are seen, other stages they're not. Okay, so but at some stage of development, each of these 4 characteristics should be present in all chordates. Okay, so dorsal hollow nerve cord here. Notochord is the support structure just ventral to the dorsal nerve cord. Then we've got the muscular postanal tail, so muscular tail that extends past the anus. And then the pharyngeal slits or clefts. Okay. So there are 3 subphyla of chordates: Urochordata, Cephalochordata, and Vertebrata. Okay. So tunicates are an example of Urochordata. We won't look at those in lab. We used to look at slides of those, but they were not very good. So we don't look at those anymore. We will look at an example of a Cephalochordata. The example is the Amphioxus, sometimes also called the lancelet. And then the vertebrates, which include the lamprey, the perch, as well as ourselves, okay, are all vertebrate chordates. All right, so let's look first at our lancelet or Amphioxus, okay? So it's an invertebrate chordate. It has all the features of a chordate, but it's not a vertebrate, okay? It is a filter feeder. It lives in coastal waters. It kind of like burrows its back end into the sediment, and then it's a filter feeder. So it just kind of allows for food to wash over it and is grabbed by its mouth parts, which you'll see as we continue looking at this organism. Um, like I said, it's in the subphylum Cephalochordata, which is named for the fact that the notochord, chordata, extends into the head, cephalo. Okay. So the notochord extends the entire way into the head, whereas in other organisms, it might stop prior to the head because that organism has a more developed brain. Okay. Which would kind of take the place, and then the notochord will develop into structures that are more support like posterior to that. Okay. And amphioxus itself, I just like to take apart the words and it helps me to remember which one's which. Amphioxus means both ends pointed or sharp at both ends. As you can see in the organism picture here, okay, it looks pretty sharp, pretty pointed at both ends. It's not actually sharp to the touch, but pointed on both ends. So this is the first organism we're going to look at, the lancelet and amphioxus. And everything that we look at for this guy primarily is going to be in slides. Okay. So this is, I have 3 videos of the amphioxus posted on Canvas. One is a whole mount, which looks a lot like this. And then there are 2 that are cross sections, just showing you one has all the anatomy and it happened to be a female. And then I had a third one that I wanted to show you because it was a male and you could see the anatomical differences. Okay, so this is a lateral view, the whole mount. This is the head end. This is the tail end of the amphioxus. So we're going to look at the mouth parts in particular. You've got the oral cirri here, the rostrum. We've got the pharynx with the gill bars and gill slits. And then just sort of behind that is the hepatic cecum. Okay, and that has a role in digestion. The atrium is this space where water will pass through. As water and food particles are taken into the oral cavity, then the water passes through the atrium and exits out the atriopore, versus food passing through similarly, but then going through the intestine, and then waste products will exit out the anus. Okay. All right, and you can see the notochord nicely here. And then we also will look for a dorsal nerve cord. And you can see that a little bit better in this lower picture. So the notochord is more pale. In this particular picture, the dorsal nerve cord has like a pinkish pigmentation. Okay, so it runs the entire length of the body, head to tail. Okay. And the topmost part is just called the rostrum. Okay. I think that covers it for the whole mount. Make sure you watch the video, though, that I showed you of the actual slide. That's what we would have had you do if you were in lab this week. Okay. All right, now here are some cross sections. Again, I do talk about all of these features in the video, so make sure that you take a look at those. But we'll show you the head. This is the second most rostral, third most rostral, and then the most caudal section of the amphioxus. So these are the buccal cirri, the little tentacles. These are used by the amphioxus to sort of filter food, allow for things to come into the buccal cavity, and water as well as food, like I said, passes through there because these guys are filter feeders. Okay. Kind of just take stuff in, sit there and let it come, food come to them. All right, so then that's where food begins, its entrance into the amphioxus, as well as water, which will continue through the pharynx. And then the atrium is the space around that pharynx, and then we'll pass through the intestine and out. Food will exit out the anus. Water exits out the atriopore. Okay. So those are the major digestive structures. I'll also mention, you can see here in this particular cut, the hepatic cecum, which helps to process some of the smaller food, especially glycogen, and allows for digestion of that. All right. And then we have muscles, lots of layers of muscles. These are called myomeres. Okay. And they're separated by myosepta. The little divisions in between are called myosepta, and the muscles themselves are called myomeres. Okay. And then we've got our notochord, which is again running head to tail, nice round structure in the cross sections through each of our slices, each of our cuts. Okay. And then just dorsal to that, just behind that, is the nerve cord. Okay. Dorsal nerve cord here, 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 and here. Okay. So you got to think of this as a continuation, through the body. Okay. All right. And then the other feature that I didn't mention yet is the gonads. So you can see here, this one is a female. These are the gonads. They're taking up a large amount of space in the abdomen area on the ventral side of the amphioxus. So I'm going to flip to the next slide so you can compare then. The female is on the right, the male is on the left. Okay, so you should be able to see inside the ovaries. You can see there are some eggs developing, and I've enlarged that. I've shown an enlarged picture of that here so you can see a little more detail. Okay, so you should easily be able to tell the difference between the male, these are the testes, and the female, the ovaries. Okay, all right, that is it for the amphioxus. Now I am going to we're going to move on to the lamprey, which we look at in the adult as well as its larval form. Okay, so this is the adult lamprey. Okay, these guys are, I feel like they're very interesting organisms because they're kind of grossly interesting, scary in a way. As an adult, they're parasites and they live in lakes and streams, and they attach to fish, which and occasionally could attach to human if they were swimming. Now, if we were adhered to by a lamprey, we could obviously have it removed, but fish really can't do that. So they serve as a large food source for the lamprey. And the adults have really scary mouths. I feel like they've got this big buccal funnel. Okay, so this whole thing is called the buccal funnel. This is their mouth region with these really pointy, called horny teeth. Okay, and then buccal papillae, which allow for suctioning, attachment and suctioning to the prey. The teeth will really grasp in there on the prey and allow for the lamprey to really attach, really stick on there. And then it has a tongue, a raspy tongue, which allows it to like scrape away at the flesh of the fish. And a pharyngeal gland, which secretes an anticoagulant. And this keeps the prey from clotting so that the lamprey can continue to feed on its prey. So to me, that's just really kind of grossly interesting. But this obviously is the way that these organisms eat. And so this is the adult. We will also look at the larval form, as I mentioned, and that'll be on slides, but this is the adult. And it can grow up to 2 to 3 feet long. It is in the subphylum Vertebrata, so vertebrates, same as us. Class Agnatha, which just means it does not have a jaw, okay, as you can readily see. A usually means no, does not have. Natha refers to the jaw, okay, no jawless fishes, okay. So the larval form, like I said, is what we're going to look at in slides. And we pronounce that, like I said, either Amicetes or Amicoites, something like that, your guess. And it feeds on phytoplankton. So it has a developmental difference between its larval and adult forms, as well as a habitat feeding difference. Okay, so let's move to our next slide. And we had looked at the mouth parts. So the entire buccal funnel shown here is here on this guy. It's got the teeth, the raspy tongue, the buccal papillae. And then also on the top of the head, there's a nostril, okay, an opening to the pineal organ. The pineal organ for the lamprey is responsible for sensing changes of light, specifically the ratio of visible light to UV light. And this is believed to allow the lamprey to gauge its depth in water. So how much light is hitting it will allow it to make a determination about how deep it is in the water and how deep it wants to swim. All right. And then there are two eyes, one on either side of the head. There are 7 external gill slits. You can see them here on the diagram, but then also up here on this one here. You can see 1, 2, 3, 4, 5, 6, 7 external gill slits. Obviously the trunk. You've got an anterior and a posterior dorsal fin, as well as a caudal fin on the lamprey. Okay. So that's the adult. We're not dissecting this guy, so we're not looking at the internal anatomy of the adult. We'll look at a little bit more of the internal anatomy, though, in the larval form. So this is very similar to the slide that I posted on Canvas, and I do want you to take a look at that. I go through it in detail, but we'll just mention, obviously, you've got your mouth region here. You've got these oral cirri, okay, these little projections are attached to the oral hood. So this line is not really lining up as well as I would like. The cirri are these little projections here, okay? So if I can move that, I would, but I think that has already been fixed. So, okay, oral cirri are the little projections which allow it to grasp and bring food into the mouth. They're attached to the oral hood. There's obviously an eye here. There's another one on the other side of the body. It does have a brain, whereas before we hesitated to really call that nerve fiber much of a brain. At most, there was a compilation of cells that we kind of refer to as a ganglion. But now we've really got an enlarged brain, okay? And you can see that in the enlargement of this dark pigmentation here, okay? So this is its brain, which of course continues down the back and is then termed the spinal cord, okay, or the dorsal nerve cord, okay, spinal cord. And then just ventral to the spinal cord is the dark pigmented line. Just ventral to the spinal cord is the notochord, and that's this paler structure here, okay? All of this, okay, running the length of the body. Again, the lines are not where they should be. So notochord is really like this pale structure. The spinal cord is the dark pigmentation. Okay. All right, and then the pharynx is food and water are passing through. And then food will travel through the esophagus right here to the intestine right here. And waste from the food will exit the anus, which exits before the postanal tail. Okay. And then I did not mention the heart, which is present just caudal to the pharynx, or the liver, which is just caudal to the heart. Okay, so you can kind of see like this heart has like a couple little rings, little circular structures to it, and then the liver is more linear. Okay. All right, so now let's look at our cross sections. Like I said, I go into this in detail in the videos. So I'm just going to talk in detail about this first one because this first picture, I was unable to find a slide that actually showed this far rostral, okay, for the larval lamprey. So for this one, I would want to point out that we've got obviously your big oral cavity. We've got the carotid arteries, which are present bilaterally, the eyes, which would be present bilaterally if it had been cut on an even plane, but it looks like it's been cut a little catty corner. So you've got one eye, the other one would also be here. The brain is obviously taking up a good bit of the space on the dorsal side of the organism in the same position as we will see the spinal cord in later sections, okay? But you can see it's a lot larger. It's got different sections to it, different lobes, if you will. And then as we continue back, this is most towards the front, the brain, the head of the organism. As we continue back, cutting back further through the organism, we're going to see that brain turn into spinal cord, okay? And then you've got the notochord present, which wasn't there when we saw the brain. Now we've got a notochord, and of course the pharynx with gills, gill bars, and gill slits. The myomeres, muscles are still here. And then I will just quickly move through these because like I said, they're on the other video. myomeres. Spinal cord kind of flattens out as you travel back towards the tail region. The notochord, large pharynx, lots of gills. Okay. Um, Spinal cord again, notochord, intestine now. We've got kidneys and a dorsal aorta surrounded all over by myomeres. Okay. And then the last cross section we've got, still got our myomeres, got a big notochord and spinal cord. The where we had seen the dorsal aorta, now that has continued into the caudal artery. We say it continues as the caudal artery when it hits the tail region. And then that is, the function is the same. It's taking blood, which has already been oxygenated from the heart to the lower parts of the body. Okay. And then that blood is returned via the cardinal vein. Okay. And that's from the heart to the tail region. I'm going to skip back to slide 16 just to show you that the carotid arteries, I don't know if I mentioned their function. This is also oxygenated blood coming from the heart, but now going to the head region. Okay. So the carotid artery takes freshly oxygenated blood to the head, and then the dorsal aorta and caudal artery take that freshly oxygenated blood to the tail. Okay. And then all that blood has to return through the caudal vein from the tail. All right, so those are the cross sections of amacoetes. And we will now turn our attention to the perch. Okay, so the perch is obviously a fish. It is a ray-finned fish in the class Osteichthyes. Okay, has a bony skeleton to it now. It's a vertebrate. External anatomy that you ought to be familiar with. There are 6 sets of fins that you should know. Okay, so we've got the anterior dorsal fin, number 1. The posterior dorsal fin is number 2. The caudal fin is number 3. The anal fin is number 4. The pelvic fin is number 5. And then if you can see, 6 is right here, is our pectoral fin. And it's present on both sides of the fish. You just can't see the other one because we're not looking at it. These all have these bony rays that are right in the fins. They support the fins, and they are very sharp. So you would, hopefully when you return to school, you can feel those and really see how they have that bony structure to them. Okay, support structure to them. All right, and then there's a little bit more external anatomy in addition to the fins. The operculum is number 7 here. Okay, it's a little flap that lies on top of the opening to the gills. Okay, so there's like a little slit in there covered by the operculum. Okay, and you can kind of see it right here. Okay. The lateral line, it's a little hard to visualize. I'm going to trace it with my pointer. So you can see there's like a pale set of dots. It forms like a line that runs down the side, and you can kind of see it right in here. That is responsible for helping the fish detect vibrations in the water. So detect movement. Obviously, this could help it sense if there's a predator coming or prey that it wants to chase after. So vibrations in the water are sensed, movement of the water is sensed by that lateral line. Okay. And then number 9 and number 10 are the upper and lower jaws, respectively. So the maxilla is the upper and the mandible is the lower jaw. Okay, jawbone. And that's it for external anatomy. So we're going to move to the next slide where we're going to look at some internal anatomy. Okay, so this is the dissection that you would do in lab this week. We have you cut from the anus along the midline and then up the operculum, and then also do the same up and across to expose the internal organs on the ventral side of the perch. Okay. All right, so you should be able to expose all of these if you were to cut, as I'd mentioned. And then the second picture will be after having removed some of these structures. So let me talk about these first. The gonads are going to take up a large amount of the room in the ventral side of the perch. Okay, the abdomen has a lot of space taken up by gonads. This one appears to me to be a female. So these are ovaries. The way to tell the difference is that the female, first of all, the ovaries have a more orange tint to them than the testes do. The testes are more pale white, kind of off white, not strictly white, but more like a cream color to them. And the ovaries have a more orangey tinged color to them. Additionally, before you even cut open the perch, you can often kind of squeeze it and you'll see eggs just coming out through the pores, um, especially through the anus. And there's, if you were to cut it open and you squish this, it's very particulate. There's a lot of particles, and those are the eggs. So if you feel the texture and look at the color, you can usually tell the difference between the ovaries and the testes. So you'll know if you have a male or female. All right, so that's taking up a large amount of the room there. The gills and gill filaments are going to be right underneath the operculum. The spleen is smaller. It's usually a darker color, and it is very much ventrally located. So it's smaller, but it's got a pigmentation to it. And its location should tell you that it is the spleen. And then the liver is usually pretty obvious too. It's a flapped bilobed structure, usually pale in color. It's very thin. So if I were to put my pointer or my tweezers underneath this, it's just kind of a flap. Okay. So you can remove all of those. If you remove the gill, the liver, the spleen, the gonads, then you'd see something like this picture down below. Okay. And what we're looking at now is a lot of the digestive structures for the perch. Okay. So the esophagus is here. It's a cream colored tube. Okay. Which is going to connect the mouth with the stomach. Okay. And the stomach is going to be located here. And then there's also usually little tube-like structures coming off of that. Those are the pyloric ceca. They secrete digestive enzymes to help with food digestion for the stomach. Okay. And then there's also the intestines. You can see the large intestine traveling here, which will connect the stomach to the anus. Okay. The one thing I did not mention yet is the swim bladder. That's this structure here. Okay. And if you watch the video that Jen made, and if you are able to do this in person, if you feel the texture of the swim bladder, it is very thin. It's just a thin little membrane that holds water and air. And this allows for the fish to adjust its location in the water. So if it has more air, it's going to be more buoyant. If it has more water, it's going to be less buoyant. And so it can adjust its depth in water, okay, using its swim bladder. All right, we are coming down the home stretch. If you then delve a little deeper, you will see a little bit more internal anatomy, okay? So we would like you to try to locate the heart, which it's ventrally located and more rostral to most of the digestive structures. So this is the picture here. It is a two-chambered heart, meaning it only has one atrium and one ventricle. Okay, slightly different from ours. And then if you were to cut through the midline of the perch on the dorsal side, you would see something like this. So this is our whole perch here, okay? The head region. So we're looking at it down from above, okay? And the top part of the skin, scales and everything have been removed and cut up towards the rostrum. And there's a lot of bony coverings in there. You kind of got to dig through to locate the brain, okay? And you'll see there's these lobes here, 4 lobes, which make up the perch's brain. All right, I believe that is it. That is it for this week. But absolutely do reference the other videos. Okay, so there's 5, I believe, different videos for the slides that you should look at. this week and 2 different dissection videos. Okay. So spend the time that you would in lab looking at those. Don't hesitate to email me if you have any questions, cfry@lvc.edu. Okay. And I will be on office hours 12 to 1 on the Zoom link Tuesday. Okay. All right. I hope you're having a good week and I will see you later.